go ahead and get started. Thanks everyone for being here this morning, March 18th edition of Nephrology Grand Rounds at University of Washington. And we have three speakers this morning. In the first slot, Dr. Lidgard will present, followed by Dr. Zelnick, and then our own Dr. Shanklin, who needs no introduction, will wrap things up at the end. So without further ado, Ben, go ahead. You're muted right now.
Can you hear me now?
Yep, there you go.
Okay, great. So just going to share a portion of my screen. Hopefully you can see the slides and not the notes.
Yep, it looks good.
Excellent. So we're going to go ahead and get started here. Thank you all for coming to Grand Rounds today. I'm excited to share some information about a very interesting area that I've been learning about over the past few months. Some of you are already aware of my interest in the area. So for those of you that are not, this will serve as an introduction to the topic, and hopefully I'll be able to convince you of its worth in the coming 30 minutes or so. So I'll be talking briefly about lipidomics, this specifically as a tool which has the potential to deepen our understanding of physiology and to potentially identify novel targets to reduce the cardiovascular risk in patients with chronic kidney disease. So just as a brief overview, we'll be focusing on the following areas today. First, we'll discuss the terminology, biology, biochemistry, organic chemistry of lipids, so we're all on the same page and know what we're talking about. Next, we'll spend some time going over how lipidomics are done, the methods. Third, we'll review some of the pertinent existing literature in the areas of chronic kidney disease and also in the areas of alterations in cardiovascular disease risk. And from there, we'll kind of delve into the unknowns and what I plan to do about those. So unfortunately, the nomenclature in the lipidome is a little bit confusing. When we as clinicians think of lipids, we're thinking of triglycerides, HDL, LDL. Only one of those is a lipid. So I want to spend the first chunk of time going over the terminology today, so we all know what we're talking about. So first off, a lipid is any macrobiomolecule that's soluble in nonpolar solvents. Translated into English, this means that lipids don't mix well with water. That's because if we consider this fatty acid here down at the bottom of the screen, you'll notice that this is primarily hydrogens and carbons. It's a long chain hydrocarbon. There are a couple of oxygens here, but there's not a whole ton of free electrons that would lend this to being very water soluble. Remember, water is very polar. So, this is not a very polar soluble molecule. However, we do need lipids for a variety of roles in the body. Little things like forming cell membranes, energy storage, hormone signaling, little roles like that are very important to us organic beings. Taking just one of these roles, let's consider the cell membrane for a moment. And I think we all remember this from high school biology. As a reminder, the cell membrane is composed of countless phospholipid molecules, and we'll consider one here in greater detail. So each one of these phospholipids in the cell membrane has this hydrophilic head group, which is composed mostly of phosphorus and oxygen, and a hydrophobic tail of varying length, which is mostly hydrocarbons. So there are polar groups facing outward and inward in the cell membrane, meaning that the interior and exterior environments of the cell are going to be very water soluble. But there's a little lipid core running through the middle of every cell wall that makes it very hard for things to pass through without specific channels. So it's not just phospholipids and fatty acids. This is a representation of some of the incredible diversity in the kingdom of lipids. There's a little bit of overlap in between the classes, but in general, you can fall into a few broad groups. There's fatty acids, triglycerides, steroids, sphingolipids, and glycerophospholipids. And the two molecules on the right here, notice these groups in red contain phosphorus and oxygen. So these are sphingomyelin and phosphatidylserine. These are examples of phospholipids. So basically they have that nice big hydrophilic head group and a hydrophobic tail. So when I say lipidomics, this is what we're measuring. We're measuring levels of individual lipid species, not HDL, not LDL. Here we're going to discuss the nomenclature, and this goes back to organic chemistry a bit. So on the left, we've got a fatty acid. This might be the simplest, least complex lipid out there. The nomenclature really depends primarily on the number of carbons and the number of double bonds. I've done the counting for you. You don't have to look that hard at the screen this early in the morning. So there's 18 carbons in this molecule. And if you want to get a little bit more granular, there's a double bond between carbon 9 and carbon 10. So we'd call this fatty acid 18,1N9, denoting 18 carbons, one double bond, and that double bond is in the 9th carbon position. That gets a little bit cumbersome. So in some cases, we have shorthand names. This acid in particular is called oleic acid. This is just the tip of the iceberg. The lipids just get more complex from here. If we add a nitrogen to the oxygen and another OH group, you can get sphingosine from this fatty acid. Further adding a fatty acid residue to that nitrogen gets you a ceramide. If you then attach a phosphocholine or a phosphoethanolamine group to the oxygen of the carbon 1 position, you get a sphingomyelin from that ceramide. And again, the number of carbons and double bonds is going to determine the name. So this ceramide here is actually ceramide 18,1. Getting a little bit more complex, we can add a fatty acid to the ceramide. Remember that R in these molecules just denotes there's some length of carbon chain there. So here, the ceramide that we've talked about before becomes ceramide 18,1 for the backbone, 18,1 for the R chain. Since the 18,1 backbone is the most common among ceramides, we'll sometimes just abbreviate this ceramide 18,1. If this was a couple of carbons shorter in the R chain, this would become ceramide 18,1, 16,1, or ceramide 16,1 for short. So since lipids are such an important part of we water-based life forms, biology had to evolve mechanisms to transport them throughout our water-soluble world. The solution that we living beings came up with is to use lipoproteins. These are little hydrophobic bubbles in an otherwise very hydrophilic world. These include HDL, which I've shown here, LDL, chylomicrons, VLDL, etc. So looking at the phospholipid layer here, we notice that there's just one layer to the envelope. Instead of a phospholipid bilayer, we now have a phospholipid monolayer. That means that the core of each one of these lipoproteins facing in towards these long hydrophobic long carbon chains is going to be very lipid soluble. There's no water in the middle of the lipoproteins. That means that there's lipid cargo like triglycerides and esterified cholesterols that are hanging out in the middle of each one of these LDLs, VLDLs, IDLs. However, the exterior of this is still a very hydrophobic solution. That's only part of the story, of course. Lipoprotein means there's some lipid component and some protein component. And we see here the apolipoproteins, again, nomenclature is confusing, are going to be clumped up on the surface of these lipoproteins. These include things like ApoA1, ApoA2, ApoB48. These have various roles throughout the lipoproteins, but they mostly are used for stabilizing the superstructure of the lipoprotein bubble, as well as acting as ligands for receptors, so they can bind to their cells and carry out their various physiologic duties. There's other classes of proteins here too, things like LCAT, PON1 through 3. These are lipoprotein-bound enzymes that often facilitate enzymatic reactions necessary for the function of the various lipoproteins. So to review the terminology, we've got lipoproteins such as HDL, IDL, LDL, and these are composed of apolipoproteins, which are the proteins kind of on the superstructure of the lipoprotein, helping to maintain the shape, and associated enzymatic proteins. And then you have all the lipids, which can either be part of the lipid monolayer envelope, as in the case of phospholipids and unesterified cholesterols, or these can be cargo, as in the case of triglycerides and esterified cholesterols. So the question becomes, how do we get these out of the patient? So obviously we need serum lipid levels for a lot of these experiments. So first off, you need to draw blood. That's where we're going to find all these levels. So first, the most important step is to draw blood. And then you take that blood to the centrifuge, you spin it down a couple times, and you can get all the cellular components separated out. You take the serum supernatant off of that, and then this contains all of these proteins like albumin that are normally in the serum. It also contains all of those lipoproteins that we want to think about. So then we go into a round of basically enzymatic digestion. Chop these up really fine, lots of enzymatic reagents, just chop up everything, every single little thing in there. And then you take it back to the centrifuge, spin it down again. Everything that's polar will be kind of on the bottom of the sample. Everything that's lipid or fat-based is going to float to the top, kind of a classic oil and water experiment. Once you pipette off that lipid layer, that top layer there, you can take it directly to the LCMS or gas chromatography mass spectrometry machine. So liquid or gas chromatography mass spectrometer, and then you can get the mass and charge of the molecular species that are present in that sample. Once you have a list of the peak list, once you have the MZ values from the mass spec run, essentially, you can literally upload this wholesale to lipidmaps.org. Now, this is a free collaborative project hosted online. I think the actual server farm is located at UCSD, but other than that, this is free. Anyone can use it, so everyone in the lipids realm does use it. Basically, this will tell you what lipids correspond to what peak list you have, what class they're in. It'll give you molecular diagrams if you want them of these various lipids. Really powerful tool, really great resource. So almost without variation, all of these lipidomics are done using mass spec. However, there's a couple of main approaches. There's the so-called shotgun or untargeted lipidomics and the more targeted lipidomics. Now, typically, untargeted lipidomics are used to identify markers and trends of interest, which are later confirmed by targeted approaches. For the targeted approaches to work, you really need to determine your calibration and internal standards, and that's not feasible with large, like n equals 100 species of lipids, just as an a priori approach. So as a result, most of the work in lipidomics and CKD so far has been untargeted. There are a few small studies out there that have done more targeted approaches, but for the most part, we're still in the discovery phase in CKD. So this approach is great, but there are some limitations that we should talk about. First, we can't tell where the double bonds in these lipids are. So this is ceramide 18-121. So we've got a standard ceramide backbone, 18-1, and an R chain consisting of 20 carbons with one double bond. So these are technically two different molecules. The Where's Waldo enthusiasts among you will have already noticed this double bond is in a different place in the R chain. But the LCMS machine cannot tell these two apart. It'll just tell you there's this many carbons, there's this many double bonds. Great, moving on to the next molecule. So it's interesting that these are two different things. Fortunately, we don't know if there's a difference between the functional significance of these two in biology. So it's kind of a moot academic point. However, the LCMS can't also tell us the stoichiometry of the double bond, whether that's cis or trans. We also don't know how many carbons are in which R chain or where the double bonds are in that R chain. So LCMS has some limitations and drawbacks, but it's still kind of the best we have for right now. There has been a good amount of work on lipoprotein levels in CKD patients, as you all are no doubt aware. So this is a really nice review by Dr. Vaziri out of UC Irvine. And as summarized here, as CKD progresses, we tend to see more LDL, more triglycerides in total, and a little bit less HDL overall. Furthermore, the HDL that we do see tends to be dysfunctional. It doesn't seem to have the same anti-inflammatory, antioxidant properties that we usually attribute to HDL, and its role in cholesterol efflux seems to be impaired. So the biological mechanisms underpinning these changes have been incompletely described. Lipidomics, however, lets us go beyond these kind of big picture lipoprotein studies and lets us actually characterize the lipid species seen in patients with chronic kidney disease, which might help to explain some of these functional changes. So this figure was done from a study by Farsad Afshinia and Subramanian Penether at the University of Michigan. They're kind of the one lab that's actually doing good big lipid studies in CKD patients. They characterized the lipid alterations in patients with chronic kidney disease in the C-probe and CRIC cohorts. So we see here that as compared with chronic kidney disease stage 2, patients with chronic kidney disease stage 5 tend to have longer and more highly unsaturated R chains present in their triglyceride molecules. Though the mechanisms are, again, a little bit unclear, it seems that chronic kidney disease might impair beta oxidation at the mitochondrial membrane. If you remember from biochemistry, this is a process by which long-chain fatty acids are broken down to intermediate length chains, basically 2 carbons, and passed through into the mitochondria to undergo energy formation processes there. So if you don't have that process, if that process is impaired, you'll see a buildup of predominantly longer hydrocarbon chains. So I've extrapolated this figure from a variety of studies across chronic kidney disease and end-stage kidney disease cohorts. So these are with and without control groups. These are not from single cohorts. They do not have the same calibration standards. You should take this with a massive grain of salt. However, it seems that as chronic kidney disease progresses, we tend to see an increase in the amount of saturated fatty acids, a decrease in the amount of polyunsaturated fatty acids, possibly an increase in the amount of ceramides and triglycerides. There's other changes too. We talked about beta oxidation, functional changes, but we might also see changes in the amount of acyl carnitines present. So far I've nerded out. I've brought up some very vivid memories from your organic and biochemistry classes from undergrad. But the important part here is that these lipids and alterations in the lipids can have very important implications for health. So the figure on the left demonstrates data from lipidomics done in the ADVANCE trial. So we can see here that phosphatidylcholines with 0 or 1 double bond tend to be associated with an increased risk of cardiovascular disease or death. The few species here that have more, e.g. 5 or 6 double bonds, e.g. they are more polyunsaturated, they tend to be negatively associated with these outcomes. Further, the data on the right are from the lipid study and show that several phosphatidylcholines with greater than or equal to 4 double bonds, again, negatively associated with cardiovascular disease or death, whereas phosphatidylcholines with a lower number of double bonds tend to be positively associated with these bad outcomes. This work was done here at the University of Washington by Rosa Lemaitre and Andy Hoofnagel with some others. So they measured the levels of ceramides and sphingomyelins in the cardiovascular health study, and they found that short chain containing ceramides and sphingomyelins were significantly associated with the risk of heart failure, whereas longer chain, e.g. sphingomyelin 20 through 24, were negatively associated with the risk of heart failure. Using the same cohort and the same sphingomyelin data, they then repeated this study with atrial fibrillation as their outcome, and again found a strong positive correlation between shorter chain ceramides and sphingomyelins and risk of atrial fibrillation, and a negative correlation between longer chain ceramide sphingomyelins with risk of atrial fibrillation. These are from a meta-analysis of studies just of ceramides. This was published about 2 years ago. And these are all in the general population. However, the authors did find a positive association between ceramide 16-0 and cardiovascular events, both as the composite of cardiovascular death and major adverse cardiac outcomes or events, and each individually. This trend was repeated with ceramide 18-0 and ceramide 24-1. So 24-1 here kind of bucks the trend of longer ceramides being better. It seems that there's something about that double bond that is not very good for health in general. Now, there are multiple proposed mechanisms by which sphingomyelins and ceramides might cause these poor outcomes. The one that's been the best described in the literature has been apoptosis. Unfortunately, there's not a whole lot of literature to suggest why longer chains should be differential risk than shorter chains. So here we're just focusing on total sphingomyelin and ceramide levels and how they might induce apoptosis. So first, both extrinsically and intrinsically, sphingomyelins can be converted to ceramides via sphingomyelinase. It's thought that ceramide-enriched signaling platforms on the cell membrane can release caspase 8, which will then induce apoptosis via the caspase 3 pathway. Further, intrinsic ceramides might induce apoptosis via mitochondrial membrane instability, which causes cytochrome C release and activation of the caspase 9 cascade. It might also cause activation of CRAF, which removes the AKT-PKB inhibition of caspase 9 and the caspase 3 apoptosis pathway, or it also might act through the PP2A BCL2 bad pro-apoptotic pathway. So essentially, all of these processes are thought to be going on with higher levels of ceramides or sphingomyelins in beta islet cells and cardiomyocytes, which might explain some of the increased risk of diabetes or cardiac disease that we see in these patients. So I presented a lot of data here. I really want to stress at this point that there's a ton that's not known previously. So as I alluded to, most of these studies investigate just chronic kidney disease or end-stage kidney disease. they really don't compare the two. We're not sure if there's functional differences between these two groups. Further, while it certainly stands to reason that these lipidomic alterations seen in patients with chronic kidney disease might be associated with an increased risk of cardiovascular disease and death, no study to our knowledge has investigated this thus far. Further, it's not clear whether dietary or medical interventions could alter the lipidomic profiles in chronic kidney disease or somehow ameliorate its effects or potential effects on cardiovascular disease and death. For instance, we know that statins really don't seem to work in patients with end-stage kidney disease on dialysis, but we're not sure if that's because of a lipidomic mechanism, a lipoprotein mechanism. There's just a lot that's not known. So it turns out that we actually do have some data on this area. So back in about 2015 or 2016, Andy Hufnagel isolated serum HDL from participants in the Seattle Kidney Study and the hemodialysis fistula maturation study. And after isolating the HDL and digesting it, they ran mass spectroscopy proteomics and targeted lipidomics, looking at 37 species in 5 different classes. None of these data have been published, so we plan on 3 main projects to use using these data. First, I'm going to run a descriptive paper. How do HDL lipidomics differ between patients with mild moderate CKD, severe CKD, and kidney failure on dialysis? I want to look at whether these HDL lipidomic alterations in patients with chronic kidney disease are associated with a greater risk of cardiovascular events. And I also want to see if HDL lipidomic alterations in patients with late-stage chronic kidney disease are associated with greater risk of failed fistula maturation. So wrapping up, we've discussed our terminology. We've gone over organic chemistry and biology. We've discussed the roles of lipids in the body. We've talked about the methods that we use to determine lipid levels. We've talked about lipidomic studies, both in chronic kidney disease and the general population as markers of cardiovascular disease risk. And we've discussed some of the many, many unknowns in this field and some of the questions that I plan to address moving forward. So in closing, I'd like to thank Brian Kestenbaum and Andy Hufnagel. They are the ones that originally measured these SKS data, and they've graciously allowed me to use them. They're also currently enduring a nonstop barrage of emails from me about the data, so thank you again for your patience there. I'd also like to thank Roseanne from the Division of General Internal Medicine, a great expert on sphingomyelins and ceramides, and a great resource for these studies. Of course, I need to thank Nisha and Leela, as always, for all their mentoring and help. And thank you, all of you at the KRI, for letting me bounce ideas off of you and talk to you about this, and for sitting patiently through this very dry review. At this point, I'd like to thank you all for coming and ask if anyone has any questions.
Thank you so much, Ben, for presenting your ideas and this overview. Definitely got me thinking to my undergraduate days. Also, while we're waiting for people to ask questions, congrats again. I think you had a first author paper just published online this morning in CJSON with our second speaker, Dr. Zelnick, as the second author. So congratulations. Ian has a question.
And not just that, if I could add, it's the lead story of the ASN in the Loop.
That's right. Congrats, Ben. Ian, go ahead.
Congrats, Ben, on the very nice paper and nice talk. It's great to see the grounding in biology here. One of the confusing things about lipids and lipoproteins to me are the various kind of axes of evaluation that you talked about. There's the various types of lipids. They're the particles in which they're packed. They're the proteins that go on them that we sometimes measure. So my question is about how do these circulating abnormalities in sphingolipids and phosphatidylcholine and things, how do they relate to the particles that you mentioned, like the VLDL? Is there an explanation in terms of particles that correlates? Because they're not flowing free, of course. We break them up before measuring them. Where were they, and how does that relate to the underlying movement and biology of these things? Or is that something we don't know?
Yeah, that's a great question. There's a lot that's not really known. Most of these studies that look at different lipid levels in patients with chronic kidney disease tend to isolate them from serum, but there have been a couple of studies that have looked, I think, at kidney autopsy or necropsy, both in, I think they're mostly in animal models, but they do show a little bit of lipidomic alterations there as well. So it's mostly from the serum. I think there are some studies showing intracellular alterations in the lipidome. But it's thought that, potentially alterations in the cargo, obviously if you get changes in your triglycerides, those can be delivered into cells via LDL, or if you have different changes that in the lipids that could induce changes in HDL, that could explain some of the dysfunctionality of HDL that we see in chronic kidney disease. So a lot that's not known, a lot of possible potential theorized mechanisms.
Yoshio has a question. Go ahead, Yoshio.
Yeah, Ben, great talk. And kind of building on what Ian was asking, I mean, you've described the lipidome as a pretty diverse, right, heterogeneous and sometimes dysfunctional entity. Like, I guess, sort of going beyond the associations, like, how do you actually map them to functional pathways in like kidney injury, or how do people map them to in different types of kidney injury?
So you were cutting out a little bit there at the end, but if I understand correctly, your question is how do you map these different lipid species to functional pathways? Okay, so a lot of this is with in vivo or in vitro to start off with basic science models. So they've taken like various cell lines from embryonic lines in mice and rats, and then exposed them to higher levels of ceramides and seen, using various labeling, whether they can induce apoptotic changes, and then the mechanisms are kind of theorized. There are some studies out there where they'll look at using like radioactive labeled sphingomyelins and ceramides. They'll get those into cells and see where they cluster. So if they're clustering by mitochondria or by the endothelial reticulum, that might point to where these things act predominantly. And it seems like with ceramides, there's some clustering on the cell membrane, but there's also quite a lot of clustering around the mitochondria in these radiotracer studies. So that seems to suggest that there's a large kind of mitochondrial component to this.
So Ben, this is Jonathan. A question for you. What do we know, or what's known about the post-translational modification of lipids as opposed to, let's say, proteins? For example, carbamylation, glycation, glyoxidation, oxidation, which we know with proteins in kidney disease. There's a lot of, and we know LDL, for example, gets carbamylated due to non-enzymatic reaction with urea. So what's known about the frequency of post-translational modification, what lipids can be modified, and what might be the downstream effects from a functional standpoint? Is much known about that?
Yeah, that's a great question, and something that I really didn't touch on much in this talk. But obviously, chronic kidney disease can induce changes in some of these enzymes or non-enzymatic processes just via differential levels of these components that are out there in the blood. So it's thought in a couple papers that you might see an increased glyceration, so sugar binding of these lipids to form like glycerophospholipids or glucosylceramides in patients with chronic kidney disease relative to the general population. The mechanisms for that are not exactly well defined from the papers that I've been reading.
Like a comment here from a question from Kim in the chat. Have you considered looking at microparticle lipid composition? We need to isolate microparticles from blood prior to digesting total serum.
Yeah, Kim, I think that's a great question. I'm interested in looking at whole serum, obviously lipidomics, but also at LDL, VLDL, remnants. I think that all of these potential spaces will contain very interesting information. So I think that microparticles are a great way to go. There's a lot of interesting work to be done here. So far, we don't really have that data available, but I think that might be a very interesting next area to think about.
All right. Well, thanks so much, Ben. And also a personal thanks from me for leaving plenty of time for questions, which not all of our speakers do. So thanks for that. And we'll move on now.